Hello everyone, my name is Visalhounuo. In this video, I'll be explaining on preparation of culture media. This culture media is a growth medium used to grow microorganisms like the bacteria and fungi, or in other words, it contains everything that the microorganisms need to grow outside the body and under laboratory conditions. The culture media which are used for growing bacteria are called bacteriological media. This bacterial culture media is used when a specific bacterium must be grown in order to confirm the presence of an infection or study a specific bacterium further. There are thousands of different media used today. It is through the use of a bacterial culture media that the presence of a bacterial infection is either confirmed or not. Although there are other ways of doing so, growing microbes is the most accurate method. In simple words, unless microbiological media existed, actually confirming a disease would be incredibly difficult. Culture media contains all the elements that most bacteria need for growth and are not selective. So they are used for the general cultivation and maintenance of bacteria kept in laboratory culture collections. Culture media are required to grow the organisms from infected material to identify the causative agents. The basic constituents of culture media are water, electrolyte, peptone, meat extract, and blood or serum agar. Based on the nutritional factors, culture media are of following types. First, simple media. Example is nutrient broth, which is a simple medium. It contains peptone water and meat extract 1%. When glucose is added to this nutrient broth, it becomes glucose broth. And when 2 to 3% agar is added to nutrient broth, it becomes nutrient agar. This is the simplest and routinely employed medium in laboratory for diagnostic purposes. If concentration of nutrient agar is reduced, semi-solid medium is obtained, which enables motile bacteria to spread. The second is synthetic media. These are prepared from pure chemicals and the exact composition of the medium is known. These are used for spatial studies. The next is spatial media. In this it includes enriched media. The media are enriched usually by adding blood, serum or egg. Examples are enriched media are blood agar and Lowenson-Jensen media. Next is enrichment media. It contains the nutrients required to support the growth of a wide variety of organisms, including some fastidious ones. They are commonly used to grow as many different types of microbes as are present in the specimen. Example is alkaline peptone water. Next is selective media. These selective media are used for the growth of only selected microorganisms. Example, DCA, which is deoxycholate citrate agar, which is selectively used for the growth of Salmonella. Next is differential media. Differential media or indicator media distinguish one microorganism type from other growing on the same medium. This type of media uses the biochemical characteristic of a microorganisms growing in the presence of specific nutrients or indicator, such as neutral red or phenol red indicator, added to the medium to visibly indicate the defining characteristic of a microorganisms. Example of this differential media is McConkey agar, where it differentiates bacteria based on lactose fermenter or non-lactose fermenter bacteria. The next is indicator media. An indicator media is included in the medium. A particular organism causes change in the indicator. Example of this indicator media are blood agar and McConkey agar. Next is transport media. Transport media are temporary storage of specimen being transported to the laboratory for cultivation, maintain the viability of all organisms in the specimen without altering their concentration, and contained only buffer and salt. Example of this transport media are glycerol saline transport media. The requirements for these preparations are electric balance, spatula, culture media powder which is commercially available, conical flask, measuring cylinder, distilled water, autoclave, petri dish or petri plates, cotton and incubator. Coming to the procedure, wipe down lab bench carefully with disinfectant to help prevent contamination of your media because it easily gets contaminated. Next, take the culture media powder which is commercially available and then using a clean spatula, measure the required amount of culture media powder for 100 ml of preparation. After you have weighed the required amount, put the media powder into conical flask. Then take a measuring cylinder and measure about 100 ml of distilled water. After that, pour the distilled water into the conical flask containing the media powder and then mix the media gently in the conical flask so that the media powder get properly dissolved in the distilled water. Then take the cotton and plug the mouth of the conical flask. Then wrap the mouth of the conical flask with an aluminum foil. Then place inside the autoclave and then sterilize it at 121 degrees Celsius for 15 minutes. After 15 minutes, allow the autoclave to cool it down at room temperature and then gently remove the media conical flask containing the media. And then hold when the media is at room temperature, hold the media and then remove the cotton plug and aluminum foil and then sterilize the tip of the conical flask in a spirit lamp and then gently pour into the culture plates and then immediately cover the culture plates after you have poured the media into the petri plates. Then after you have finished pouring all the media into the culture plates, then to check whether their preparation has been contaminated or not contaminated, put the petri plates into the incubator and incubate it for around 18 to 24 hours. And then on the next day when you come and observe the culture media, if there is no growth, then it indicates your preparation is sterile. And you can proceed for growing the microorganisms or growing the bacteria. Coming to the uses. Culture media are required to grow the organisms from infected material to identify causative agents. Here I have described the media with their uses. In case of liquid media, first is peptone water, which is used for routine culture and sugar fermentation. Nutrient broth is used for routine culture. Glucose broth is used for blood culture. Alkaline peptone water is used for culture of Vibrio. Selenite F broth is used for culture of feces of Salmonella and Shigella. Tetrathionate broth is used for culture of feces, especially for Salmonella. Next is Robertson's cooked meat broth, which is used for culture of anaerobic bacteria. Next is under solid media, you have nutrient agar, which is used for routine culture, blood agar, which is used for routine culture, chocolate agar, which is used for culture of Neisseria and Haemophilus influenzae. Loeffler serum slop is used for culture of Corynebacterium diphtheriae. McConkey agar medium is used for culture of gram-negative bacilli, and deoxycholate citrate agar is used for culture of Salmonella and Shigella. And then bile salt agar, which is used for culture of Vibrio cholerae. These are selective media. Next, coming to the common media which are routinely used. First is nutrient broth, which contains 500 gram meat extract in 1 liter of water or distilled water. This 10 gram of peptone and 5 gram of sodium chloride are added. pH is adjusted to 7.3 and it is used as a basal media for the preparation of other media, or it is used to study soluble products of bacteria. Next is nutrient agar, which is solid at 37 degrees Celsius with the addition of 2.5% agar is added in the nutrient broth. It is heated at 100 degrees Celsius to melt the agar and then cooled. Next is peptone water, which contains 1% peptone and sodium chloride 0.5%. It is used as base for sugar media and to test indole formation. Next is blood agar, most commonly used medium, and then it contains 5 to 10% defibrinated sheep or horse blood is added to melted agar at 45 to 50 degrees Celsius. Blood acts as an enrichment material and also as an indicator. Certain bacteria, when grown in blood agar, produce hemolysis around their colonies. Certain bacteria produce no hemolysis. Types of change: beta hemolysis. The colonies is surrounded by a clear zone of complete hemolysis. Example: Streptococcus pyogenes is a beta hemolytic streptococci. Next is alpha hemolysis. The colony is surrounded by a zone of greenish discoloration due to formation of biliverdin. Example: Viridans streptococci. Next is gamma hemolysis, where no hemolysis is seen and there is no change in the medium surrounding the colony. Next is chocolate agar. Chocolate agar is prepared, which contains heated blood agar. Prepared by heating blood agar, it is used for culture of pneumococcus, gonococcus, meningococcus, and haemophilus. Heating the blood inactivates inhibitor of growth. Next is McConkey agar, which is one of the most commonly used for Enterobacteriaceae. It contains agar, peptone, sodium chloride, bile salt, lactose, and neutral red. It is selective and indicator media.